Super.
All right, so as the Zoom platform just informed us, this meeting is being recorded, just so everyone knows. So if you don't want your video on or your mic on, go ahead and turn those off now. But I'm really excited actually to introduce our speaker today, Erin Powers. Erin's one of my friends, so be nice, but she doesn't. Erin is going to talk to us about dental microwear in Romans, and she's currently a staff archaeologist at Ball State University in Indiana. She has loads of ongoing projects, but one of the coolest ones is a geophysical survey looking for the cemetery of the first psychiatric hospital in Indiana. So if you have questions about that at the end, I'm sure she'd be happy to talk about those too. But first, we're going to learn about dental microwear. So over to you, Erin.
Yay! Thank you so much. Hi, everyone. Thank you for coming and tuning in today. I hope you guys are staying safe and healthy across the pond. And to some of the students that I saw here that chimed in, thank you, Kate, and the Department of Archaeology and Conservation at Cardiff for inviting me to present. And I hope you guys find this as interesting as me and Kate do. So the way I'm structuring this presentation is I'll give about a 30-40 minute presentation, and then at the end we should have time for questions and any comments that you have. I'll also answer questions about the most recent season of Love Island UK, but please wait until the end for those as well. So like Kate said, I am Erin Powers, and I'm an archaeologist here in the Applied Anthropology labs at Ball State University. And we're in Muncie, Indiana, which is actually the home of Jim Davis, the cartoonist who created Garfield. And that's about it. So now you know everything about Muncie, Indiana. That kind of covers it. So I'm going to talk to you about dental microwear texture analysis. We're going to talk about what that is, how it's used, and then we'll examine a case study that involves dental microwear texture analysis, but also called DMTA. So the case study that I'm going to present to you is the DMTA of individuals from 2 Roman period sites, one in Romania and one in Italy. Okay, so I've broken this presentation into 3 sections. So I'm going to give y'all a brief history of DMTA, how it started, and how it grew into a super useful scientific method in the bioarchaeology or osteological world. Then I'm going to talk about the methodology behind DMTA and basically how it works. And then finally, we're going to look at a cool case study of how DMTA has enhanced our understanding of dietary habits during the Roman period. Oh shoot, you're on the wrong screen.
Yeah, we just see the, we don't see the presentation view,
Oh,
we're seeing the notes view.
Oh, that's so lame. I'm so sorry. Okay, that's very cool.
Believe you me, the standard of interaction I've been having with my students this week, this is stellar.
How is that? Okay, perfect.
That's fine.
Awesome. Okay, thank you. I'm glad I read that. Okay, so now that we're all on the right page and the right screen. So these are kind of the bullet points that I'm going to be going over with you guys. So let's kick off and go into a brief history of DMTA. Okay, so how did this study of teeth get its roots? Little two-stroke. So we know that teeth are an invaluable resource in the archaeological record. They're strong, they're made of mainly inorganic materials, and they're almost always recovered archaeologically. So on the left side of your screen, you'll see an individual from the Roman period necropolis in Romania, and you can see that maybe about 50% of the skeleton is complete, but it's pretty severely fragmented. But one of the remarkable things is that while the rest of the skeletal elements are fragmentary, nearly all of the mandibular dentition is still in place, and you can see that on the right-hand side. And since this is more or less typical of excavations, this is what makes studies like DMTA possible, is the fact that we truly recover so many dental remains. So before scientific studies like DMTA, archaeologists relied on historical narratives and faunal and botanical remains to provide information on ancient diets. And these studies are limited in that they tell us a lot of general information about ancient lifeways. So these studies not only suggest diet, but also subsistence, waste disposal, and modern collection of, or modern archaeological collection practices. So what people are targeting now to try to exhume. And what DMTA does is it targets microscopic features on the chewing surface or the occlusal surface of teeth to extrapolate what humans and animals are really eating. So on the screen, you'll see the same dentition from the previous slide, and the red square that's highlighting the occlusal surface of the second mandibular molar. And then that small portion, there's a small, tiny microscopic portion that gets scanned. And this is what this really colorful 3D image on the top right represents. And then the black and white 2D image of that same surface is created to show us how the microscopic features on the occlusal surface are left by food and grit that kind of gets accompanied by the food that we're chewing and eating. So before DMTA, archaeologists started to study dental morphology, like what Kate is studying, and started to try to study the mechanics of chewing. And researchers found that across species, that animals chew in almost the same way, and that this chewing happens in 3 different stages. And you can see this kind of represented on the image in your screen. So in the first stage, your mouth is open and it starts to begin to close to crush the food that's in your mouth. And first of all, your mandible will jut out laterally, and then it moves back medially into center. The second stage is the power stroke, where your teeth aren't touching because there's food in the way. And during this second stage, this is where the microfeatures form on your teeth. So these microfeatures are what's being studied in dental microwear and in DMTA studies. And then finally, in the third chewing stage, your mandible opens back up and returns to the starting position to start that chewing cycle over and over again. So this chewing stage, these chewing stages are being demonstrated by this camel right here, which actually does a really exaggerated but super accurate chewing mechanics for us. So once researchers figured out that most animals have the same chewing pattern, they began to explore if different foods leave behind different microfeatures on teeth. And this type of thinking is what led to dental microwear studies and to DMTA studies later on. So the first dental microwear studies used a scanning electron microscope to produce 2D scans of specific wear facets for microwear studies. And researchers had to originally hand count microwear features within a small microscopic portion of the occlusal surface. Dental microwear studies are still being conducted today, but not with the same amount of fervor as DMTA studies are. DMTA studies are a lot more popular nowadays, and we'll get to why. But one of the reasons for this is that DMTA studies capture 3D scans of the occlusal surface instead of 2D scans. And researchers argue that DMTA studies are more objective than just straight up dental microwear studies. And the images on this slide are of 2 dental microwear 2D scans that are taken on a scanning electron microscope. And you and I can tell by looking at these 2 scans that the microwear patterns definitely look different. On the left-hand side, you see a lot more linear features and scratches. And then on the right-hand side, you see a lot more of these big, like crater-like pit features. So you and I can tell that these are different. Now, what the issue with dental microwear studies like this is that they rely on a visual subjective researcher observation. And the difference between that and DMTA is that DMTA relies on mathematical computations that reflect these visual differences that we're able to see. So to correct the user error in dental microwear studies, there's now a software and it calculates these microwear features for you. But this is one of the nice things about DMTA is it gives you objective data that you can then use to try to figure out what's happening between populations or even between individuals. So DMTA studies can be applied to non-humans, ancient humans, and even modern living humans to understand dietary habits, basically anything that chews food. So it's a super versatile study. Some of the most famous DMTA studies look at primates or they look at hominins, and they try to understand seasonal variation in diet or reconstruct dietary habits. So the image on the screen now are scans of wear facets from Neanderthal individuals that were excavated in Croatia at the Krapina site. And the topographic 3D images to the right, those colorful images, give you an idea of the variation in depth of these features. And it's a really nice tool to be able to figure out, okay, what's the depth of features and kind of see some of the more linear features happening as well. So now y'all are caught up with the history of DMTA. So let's get into a little bit about how it actually works. So first thing, you need a skeletal assemblage and you need teeth to study. So here are examples of some of the skeletons I studied from a Roman period necropolis in Romania. Once you have your skeletons in your dental assemblage, you're going to make molds of these teeth using a really standard molding material that's similar to the material dentists use to make impressions of your teeth. And to give you an idea of how this works, this is an image of my assembly line where basically I set up all of the teeth that I needed to mold, and I pretty much went right down the line and started to mold all of the molars that I wanted to be able to study back in my laboratory. So this type of green putty material, it dries very quickly, like within a couple minutes, and the molds are incredibly resilient and they last for a very long time. So typically you want to mold the first and second molars, either mandibular or maxillary. The third molars, or your wisdom teeth that most of us have, they tend to grow in unpredictably just because of crowding and the way that modern humans grow in molars. And so it's much harder to figure out if the same wear facets on the occlusal surface of your teeth are being worn in the same pattern as the first and second molars. So it's not typically recommended to use for DMTA studies. So once you have your dental molds, you come back to the lab and you make casts of these dental molds. And these casts serve as a replica of your tooth. And now you take the tooth replica or the cast and you study that microscopically with a white light confocal profiler. So on your left is an image of what a white light confocal profiler looks like, and it is beautiful. So on your right, you'll see a close-up image of the microscope stage and the tooth replica, which is immobilized right here on this little green piece of putty so that it doesn't move around while you're trying to examine it. So when you're looking at the tooth microscopically, the goal is to capture a 3D scan of one of 3 phase 2 wear facets. And these wear facets are called 9, 10, 10x, more or less. And then there's also one called N. And you're looking for one of these 3 facets on either the maxillary portion of the molars or the mandibular portion. So where the occlusal surface is circled on the image in front of you is where these wear facets are located. When you find the area on the tooth where these wear facets are located, you'll go ahead and capture a scan, and the software that's attached to the white light confocal profiler will put out an image for you to process. So just to recap our steps, you've now molded and casted your teeth, you have scanned the tooth, and now you've created a 3D scan that shows you microwear on the tooth. And this is a screen grab of what you would look at when you're trying to find the wear facets microscopically. And if you look at it, you can see all of these tiny scratches, and there's some pits here as well. And this is microwear. So this is what we're trying to capture, and this is what we're looking at. So this is a blown up version of a 3D image that looks, and it looks like a tiny portion, or no, I'm sorry. So this is a super tiny portion of the occlusal surface. You can see that it's about 100 micrometers in length by 230 micrometers in width. So very small section of your tooth. And this 3D scan is supposed to show you the depth of the features and all the complexity of the occlusal surface. So now you're going to take the 3D scans that we've just created, and you plug them into 2 different types of programs to get your DMTA results. So this is what one of the programs looks like, and it is cleverly called ToothFrax. The end result of running this program is that you end up with data for 4 different values that mathematically explain the microfeatures that we're looking at. And these 4 values are commonly called complexity, anisotropy, textural fill volume, and scale of maximum complexity. So I'm going to try to explain these in a way, they're very abstract concepts, but I'm going to try to do my best to explain them simply. So complexity is the measure of roughness at varying scales, which then gets averaged and computes a complexity value for each individual scan. So human complexity values range from 0.5 to 3. However, a majority of individuals fall between 1 and 2. So the scale of maximum complexity is a value that's related to complexity. And this measures the roughness of the tooth, but it's not averaged and it's computed mathematically. So it gives you the maximum complexity. Anisotropy looks at the direction of linear microfeatures on a tooth surface. This image is an example of microwear linear features going in about the same direction, and this would be considered high anisotropy value. So the anisotropy values for humans tend to range between 0.005 and 0.01, while most populations span 0.002 to 0.004. Textural fill volume represents 2 components of the surface of the tooth. It represents the shape of the surface and also the texture of the surface. But most simply put, textural fill volume represents the amount of surface missing due to chewing and the enamel being worn. So the 4 DMTA values suggest what types of food people are eating, whether they're hard and brittle or tough and fibrous are typically the categories that we kind of discuss food in. These values suggest that how food is being processed as well. DMTA values can tell us if there are differences between diets and populations based on age, sex, social status, and a lot of other factors. So once you run your 3D scans through both software programs, you'll receive your results, and then it's time to analyze your data and figure out what's going on. So the top 2 images, for example, are from a known pastoral individual, and the bottom 2 individuals are from a known individual that practiced agriculture. So the big thing to note here is that the pastoralist on the top has microwear features with a really low anisotropy value. And we can tell this because these linear features are all in different directions. So there's really no There's no anisotropy going on here. Now, the agriculturalist on the bottom has a much higher anisotropy value, and you can see this because the linear features or the linear scratches are, for the most part, going in a very similar direction across the surface. So that just gives you an idea of what anisotropy values were to look like. Okay, so now you know the history behind DMTA and you have an idea of how it works. So let's apply our knowledge to a Roman period case study. Okay, so we're going to compare two Roman period skeleton assemblages or skeletal assemblages. One is from Vanieri, a site near modern Puglia in Italy, and the other is from Dedeul Furculor. It's a necropolis in modern Alba Iulia, Romania. And this is a map right here of the Roman Empire, and the different colors are to illustrate when provinces were conquered by the Romans. So this red marker towards the southern portion of Italy is about where Vanieri is located. Vanieri was a rural farming village along the Via Appia to Rome. It became an imperial estate in the 1st century AD, and most of the people living and working in the village were likely lower class citizens, but some scholars have started to propose that some wealthier citizens lived in Vanieri as well. So excavations of the Vanieri necropolis began in 2002 until 2017. A majority of the burials dated from 2nd to 3rd centuries AD, and individuals were either inhumed or cremated, and almost all of the individuals were accompanied by grave goods. Stable isotope studies on these individuals confirm that a majority of individuals were born and raised near Vanieri or somewhere in the southern Italian peninsula. Okay, so the Dedeul Furculor necropolis is located in the Roman city Apulum, which is modern day Alba Iulia, Romania, or as my husband calls it, Albus Dumbledore, Romania. Apulum was the military capital of the Dacian province, which is this more circular province right here. And it was the last province to be conquered by the Romans in the mid-2nd century AD, and it was the first to be abandoned in the mid-3rd century AD. So Romans conquered the province mainly so that they can control the salt and gold mines and also agricultural land. Apulum was home to the 13th Gemina military legion, and it had the largest military fort in the province. Apulum was actually made up of 2 towns at the time. The first was comprised of immigrants, locals, soldiers, and veterans. And then the second larger and more affluent town was where the Roman elite resided and was considered a more aristocratic city. And this aerial right here shows you the it shows you an aerial of the modern Alba Iulia. And if you look at this starburst shaped fort right here from the Renaissance period, underneath is where the original Roman fort was. The Dedeul Furculor necropolis is the 2nd largest to be excavated in Apulum, and it's located southwest of the city center, and it's highlighted by this big circle A is where it is. So now you have a sense of who these populations were and that we're going to be studying. So now let's discuss what we know about diet in Vanieri and in Apulum. So at Vanieri, animals were kind of the focus of the economy, using their wool and secondary products. Sheep were used for fur and to make cheese. Goats and cattle were used for secondary products. We also have charred plant materials recovered from Vanieri, and those include durum wheat, which was grown at the estate. There's no real evidence that wine and olive oil were being imported into Vanieri, but more than likely, Vanieri was producing enough olives and grapes at the estate to be able to produce their own. Isotope studies at Venery suggest that these individuals were consuming a C3 plant-based diet with a very marginal amount of terrestrial animal protein. So mainly grains and cereals, very little animal protein. So in Apulum, we have lots of cow and pig bones, and these are the most commonly found in the subsistence record at Roman Dacian sites, and specifically in Apulum. At a large site near the Acropolis, archaeologists found bones from cattle, sheep, goat, pig, horse, and birds, further suggesting what people were eating or what was available. Romans imported vintage wines, olive oil, and fish sauce into Dacia from all over the empire. In Apulum and in the Dacian province, salt was a super necessary tool in order to preserve produce and meats in Apulum. It's likely that salt as a method of preservation was used a lot since there were salt flats right near the city and since it was such a big export. Charred botanical remains that were discovered just outside of Apulum included emmer wheat, spelt, barley, oat, and rye. And within the same area, this millstone on the left was actually discovered. Artifacts like this millstone tell us how grains were being processed in Apulum. And this millstone was made of a gritstone, or a really coarse grained sandstone that's local to Apulum. Okay, so now knowing what we know about the history of Venery and Apulum and knowing a little bit more about their diets, I had originally hypothesized that there was not going to be a significant intrasite difference between Venery and Apulum in terms of DMTA values. But as luck would turn out, there were some differences between Venery and Apulum individuals that I studied. So let's get into what those differences were. Okay, so here is a chart that breaks down the DMTA average values for the two populations analyzed. So there was 31 individuals analyzed in Apulum and 20 individuals analyzed at Venery. And right away, you can see that the ASFC, or the complexity values, are much higher in Apulum at 2.34 than they are at Venery at 1.95. Simply, this kind of means that people sampled from Apulum were eating more brittle and hard foods than people in Venery. The Epulsar value, also known as the anisotropy value, are almost identical at both sites, so 0.0026 and 0.0025, and these were not significant. The textural fill volume, or TFV, and the RSMC, or the rank scale of maximum complexity, were both statistically significant when I ran my study, but there were some intersite differences within the populations that likely influenced this intrasite significant difference. And that's a bit beyond the scope of what we're going to discuss here. It's a lot of statistics, and you do not want me to go down that rabbit hole, I promise. So for right now, we're going to focus on what the complexity value differences are between these two sites. And in order to do that, we should compare Apulum, which is this dark blue diamond at the top of the graph, and Venery, the lavender color diamond below Apulum, to other sites with DMTA values. So this figure represents a bunch of different groups and populations across time spans and across dietary habits or subsistence habits, and it compares them to one another to try to figure out what is going on. So for example, all of these circular populations that you see on this graph, those are forager communities, and then the triangles are farming communities. And foraging populations typically have a high complexity value from eating hard, brittle items like nuts and seeds. And farming communities typically have a low complexity value, but a high anisotropy value from eating processed grains and meats, and more standard things. So what's really interesting here is that venery falls in line most with the foraging groups up at the top right here. And this would suggest that people at venery were eating hard, brittle foods more regularly. And what's even more interesting is that apulum has this crazy high complexity value. This is to the point where my professor used to joke with me that these people must have been eating rocks, is essentially how brittle and how hard their diet seems to be based on the DMTA. So what do these values mean? And what are some of our theories that would explain these results that we're seeing? So it is likely that people living at venery and apulum are consuming a varied diet of grains, terrestrial animals, nuts, seeds, spices, and produce. But there are several possibilities as to why the complexity values are different, and this includes food processing, preservation techniques, or probably both. So we know from the archaeological record that both apulum and venery are processing their grains with bulk milling technologies that were typical during this time. However, the material type of the millstones is different from site to site. So the one millstone that I showed you earlier that was discovered in apulum is made of that super abrasive sandstone, and it's likely expelling coarse grit into the grains that they're milling, and it would have ended up in foodstuffs and resulted in that increased complexity value that we are seeing. The millstone fragments that have been recovered at venery are made of a volcanic stone, and these are likely less abrasive, and they produce a much finer grit that would not contribute to a high complexity level. Okay, and then this is a picture of Monte Testaccio in Rome, and it's the hill that in Rome that's made entirely of broken Roman pottery. And this hill represents the sheer volume of storage vessels in Rome that people would use to store and preserve foods. Just so I can talk about food preservation in the Roman period. So archaeological evidence tells us that salt was a preservation tool for meats, dairy products, and vegetables. And it's very possible that salt as an abrasive causing microwear features contributed to this high complexity value that we see at apulum. So people in apulum ate more meat and animal products due to the amount of land that they had to rear animals, the super harsh winters where you would have to store food, and then the large access to salt via the salt trade that went right through apulum. So to sum up what we've discussed here, studies like DMTA are incredibly useful to reconstruct ancient human diets. It's not a destructive science, it's not terribly expensive, and it's fairly easy to be trained to collect microwear data. This study specifically is fascinating in that we're able to shed some light on the variety of diets and food processing happening within the Roman Empire. And this really contributes to the overall narrative that the empire was an incredibly complex and diverse region, and it refutes some of those antiquated notions that the empire was just one giant uniform culture, which we're really getting away from. And that brings me to the end. So thank you all for listening in, and thank you, Kate and the department, for hosting me during this super crazy time. And if you have any questions, now feel free to unmute yourself, or feel free to also write it in the chat box if that works for you.
Yes, thank you, Erin. If we could give her a round of applause. Yay.
Thanks, y'all.
We have to unmute. So I have a question, but it's a fairly obvious one. Is how much is the mic? How much is the equipment? You know, your beautiful whatever it was called.
I'm sorry, say that one more time.
how expensive is it to buy the well, it's a microscope, but it's not really a microscope, is it? That actually kind of scans. Is that a particularly expensive piece of kit?
Yes, the white light confocal profiler,
Mm.
I believe when we bought it, I want to say it was about $250,000 US.
Whoa. Okay.
Yeah, my advisor had received an NS. He received like a pretty large federal grant to be able to purchase that. And that is obviously the biggest expense. The rest of the molding materials, casting materials, are fairly inexpensive once you put in that enormous
Yeah,
kind of
yeah,
big machine.
because we've done sheep teeth, for example, using kind of the traditional techniques. Because obviously Ingrid Mainland in the UK does a lot of work looking at, basically sheep teeth. I mean, yes, the advantage for the 3D is that it's much easier to analyze, isn't it? Really, and you get much more detail. That's what Hannah says. That's why I didn't even bring it up. Hannah, we can save up. We can save up.
Hey, you can also always send your dental molds over to a lab, and they can process it for you and send you the results.
Interesting.
Yeah,
Interesting.
I think there's maybe 5 active white light confocal profilers that are dealing with DMTA studies in
Yeah.
the world, and I think most of them take they'll take samples constantly and
Okay. And so how long, sorry, I know we're talking about technical stuff here, but how long does it take to scan a sample?
Probably 3 to 5 minutes. It
Okay.
doesn't take very long.
Yeah, because we were looking at the costs. So last time we did it, we had to go somewhere else to get them covered with gold. You know what I mean? And the cost associated with that, and the time, in fact, is
Yeah.
not insubstantial. And then obviously the recording methodologies are different because you're using different sort of capture techniques. So I suppose at the other end of it is how useful are those sort of comparative ethnographical studies for kind of I suppose what humans eat? I mean, are you looking at trying to sort of work out which group they most look like, or is it just it'll be the patterns saying that A is different from B? Because I mean, the issues around that is actually quite hard to get clear patterns from people. I mean, I don't know. Do people cast their own teeth? I mean, do you have do you pursue a hunter-gatherer diet and then cast your own teeth as an example? I'm assuming these are from, extant populations or populations who you assume have those kind of subsistence strategies. So I guess the question is, how difficult is it to get more than A doesn't look like B and maybe they were eating slightly gritty food?
Yeah, so those are all really good questions. One thing to answer your question is because we're able to test living populations, there have been a lot of modern living populations that have been studied from different foraging, agricultural, or pastoral backgrounds. And so that kind of gives us a sense of what people are actively eating and then what their microwear is looking like. There have also been some other like kind of sillier studies where people change their diets or eat a certain type of food, and then over that process of 6 months, a year, 2 years, they'll continue to take samples and collect their own microwear data. So that is something that people do. And then some things that we're doing for ancient populations is just trying to collect as much data as possible from different agricultural groups, different pastoral groups, different foraging groups. And so part of that, this more or less falls into what my advisor is doing, which is just collecting this enormous database of DMTAs populations just to try to figure out what groups seem to match and what values seem to correspond best with different like subsistence strategies. But yeah, it's certainly not a perfect science, and there's always some agricultural groups that fall more in line with pastoral groups or foraging groups that look more like your agricultural groups. So that I think falls into this whole complexity of well, are they processing foods differently, or is it a certain type of, you know, local food that just like is reacting differently? So there's a lot of like underlying questions that still need to be answered.
Yeah, because I mean, I can remember, I can't, God, who wrote, years ago, I mean, there was a very early paper, wasn't there, that basically looked at hunter-gatherers versus early pastoralists, and, you know, basically said one of them had scratchier teeth because, you know, they ground things with stone, you know. But I was wondering, you know, how much, I mean, the techniques obviously moved on, but how much, you know, it's a difficult question to answer, is, are we just reaching a point where it's kind of more around population mapping and building up these giant databases to get away from what a very simple, you know, they ate gritty food, they didn't, you know.
Yeah, and that's really tough to answer, because statistically, if you're looking at, you know, in between populations, you're looking at these like incredibly small individual numbers, and so that in itself can throw off the data that you're trying to look at or the phenomenon you're trying to explain. And on the other hand, you know, looking at these large population-based intra-site differences, you're able to, you know, more or less figure out a pattern of what you're seeing. So yeah, somewhere in between is probably the perfect thing. But yeah, trying to get that and trying to explain your rationale is tough.
Yeah, but you have a much better method for getting to that than people used to have. Because yes, I was used to look at it and think, oh, come on, pits and scratches and measuring them. I mean, we've done it, but I was never convinced that it wasn't subject to so much user error and, you know, well, basically, subject to subjectivity, that anything that actually made it much more objective and rigorous, I think, would push the methodology forward. So it's good to see it. Thank you.
Yeah, absolutely. Thank you so much. Yeah, Hannah?
So this is going to be a really niche question that will make sense when you know that I worked in Harghita County, just northeast of Alba County in Romania.
Yes, Romania.
I actually originally was going to do my master's thesis on microwear and non-metric traits in secular people, and then I got fucked over by my field school. Yeah. So, okay, it's slightly off topic, but what were the rate of caries in your sample?
You know, I actually didn't look at caries rates. I can certainly get back to you on that. That's a really good question.
I'm mostly just curious because when I worked, our earlier periods were 1300s, so not Roman by any amount. But I mean, adults didn't have molars by the time they were adults, because the rate of caries in the population I worked with was just so wild. So whenever anybody works even like slightly near to where I did, I'm like, what did you have? Because it was really like, I mean, we had sub-adults that were like 14, 15 who were already at wear stages I would expect in like a 50-year-old adult, which was just not something I've ever seen again or seen anybody ever anywhere else see. So
sure, yeah. Based on, you know, my memory of trying to remember what all the individuals look like, the caries rate was pretty low. That being said, antemortem teeth loss was so prevalent, obviously, and once there's a fine line between, well, did this individual have caries, and then also, was the wear so bad that there was no enamel left? So pretty much that was more or less the answer, was yeah, there probably was like a pretty prevalent amount of caries. But because the wear rates were so high, much like what you saw in your population, I mean, by the time I was looking at people that were over 40, for example, more or less, like a lot of them just didn't have molars left, like you were just saying. That is a really good question, and I will definitely get back to you on that one.
Yeah, it was something interesting that I like to ask even like the Roman people who study in Romania, because we were never really able to pinpoint if it was the introduction of corn or not, because that was my original thought, which was what my thesis was going to be based on, is that it had to have been the introduction of corn because where else were they getting this such a high sugar diet? Meanwhile, they had like no vitamin C in their diets. I mean, like every single skeleton had something wrong with it. Every single skeleton had something wrong with it. I mean, even our like toddlers had something wrong with them. I mean, from rickets to like, you know, very clear large bone infections and things like that. I'll never be able to work on a population like that ever again, because first of all, the amount of juveniles we had was just insane. But yeah, so that's why I ask, is because we know that they all had scurvy and they all had no teeth. So it's been like this never ending, every time someone brings up Romania, I'm just like, so what did you have?
Yeah, our paleopathology was pretty low. I mean, I had expected, since this was like a high veteran military population, to have seen a lot more broken bones, to see a lot of, you know, God knows what. But I think I saw one broken forearm looking at this whole population. So I was very curious, and that's a whole study in and of itself.
I won't take up too much time because that is also weird.
I mean, yeah, the skeletal assemblages in Romania are so often not looked at, or not like super well researched, just because, you know, commercial archaeology over there is just, people are just constantly digging up things and they don't have time to study them. So yeah, studies like this study and your study for sure are, I mean, there's so much information to go through and so much is happening, and it's a crazy region.
So we have a question in the chat from Jesse.
Oh, thank you.
On the chart with the circles and the triangles representing foraging and farming, there was another farming group at the top. It was the Canterbury group, if I remember. So would that have a similar explanation with the grinding stone, or would that be an outlier? And I stopped your screen sharing for the question, so you might have to start that again.
Oh, that's okay.
Sorry.
Yeah, let me go back to that, because I'm actually pulling that up as well. Okay. Come on, buddy. Well, okay. Okay, so okay, can everyone see that?
Not yet.
No, not yet. Not yet. Not.
There we go.
Okay, super. Okay, so yep, so you saw this Canterbury, UK. So this is really interesting, and this population is a medieval population from Canterbury. And yes, you're absolutely right. It has an incredibly high complexity value, which is this AFC on this side, and it has a very low anisotropy, which means that the linear features are going in all sorts of directions, which is not typical for farmers. And then at the same time, they have this incredibly high complexity or roughness of their the occlusal surface of their teeth. Is it an outlier? No, because outliers for this, where I got this data, were thrown out. But it does bring up a lot of really interesting questions as to what is happening with this Canterbury population in the medieval period. And that is obviously something that I have not researched, but it's something that my advisor actually looked at, and I can send you the article, Jesse, that discusses, you know, why certain groups seem to be more like groups that you wouldn't assume that they would be like. Yeah, I mean, it probably comes down to maybe dietary shifts from standard farming practices that you see with all of these other groups. It could also be the way that they're processing their food. It can also certainly be the way that they're preserving their foods. So there's a lot of different answers as to why we're seeing this phenomenon, but it's definitely something like incredibly interesting, and definitely something that someone in the future should look at.
Yeah, I think we have time for one more question, Richard.
Just another methodological one. What's the sensitivity of the molding like? So how small are the smallest features that can be picked up? And is there a chance in the future that we might pick up even smaller features that result from the consumption of less abrasive foods? So what level are you at the moment? Because obviously kind of beholden to the putty to some degree, aren't you? So do you get down to sort of 10 microns, 5 microns? What sort of sensitivity like?
Yeah. So we can get down to 10 micron level seems to be is like kind of the industry standard for DMTA studies. Anything less than that, sometimes the software picks it up and the software thinks that it's seeing features that it's not. So we try not to do that. That being said, the molding material really does map almost the entire surface of your tooth. And even before we do the molding, we scrub the teeth with just a cotton swab and 95 to 100% alcohol to make sure that there's no dirt getting on the surface. Even then, microscopic dirt and dust still ends up in the molds, and that's something that you can't avoid. And we end up kind of like erasing those features and trying to make sure that all you can see is the microwear. But that is one of those tough technical things that we kind of have to work around. Is there's still dirt and there's still things that get into the mold. But as for the actual like methodology behind the molding, it's pretty good. I cannot imagine it getting better than it is.
Because probably anything smaller than that is going to end up being noise, I guess, effectively, isn't it?
Yeah, and also at that fine of a scale, you really, it's not representative.
Yeah.
And when you're looking at a wear facet that's, you know, maybe a 50 micrometer by 50 micrometer, it's okay if it's not the finest scale,
Sure.
because for this study, it's not specifically helpful, but the white light confocal profiler will do some like serious, you know, finite 5 micron, 1 micron. So if that's what you need and that's what you want, it'll do it.
Thanks very much.
Yeah, thank you.
Great, and we're about at an hour, so I think it's time to let Erin go. But just another round of applause. Thank you so much for the crash course in DMTA. I know that there's master students here, so I think that we might have some project proposals soon.
It's super fun. Yeah, please, Kate, if anyone wants to reach out to me, like, please email me. I love talking about this stuff. It has been such a joy. I find it super fascinating. If you need anything, you know, let me know, and I will certainly do my best.
Yeah, and we should say that it's, well, 11 o'clock now where Erin is, so this is the middle of her day. She's taking time out to be with us, so thank you.
It's been fun.
Thank you, Erin.
Yeah,
Yes, expect some parcels in the post.
Yeah. All right, thank you, everyone.